Just testing sound. Can you guys hear me okay? So hopefully people on the webinar can hear me. And I'm really excited to welcome you all to the NOAA Glaral Sigler Great Lakes Seminar Series. My name is Kiata Zuccarino-Crow. I'm with Sea Grant. And I have the honor today of introducing our speaker. Dr. Maureen Coleman is an assistant professor in the Department of Geophysical Sciences at the University of Chicago. She's a microbial ecologist and is currently leading a lab that's characterizing microbial communities, genomic diversity, and biogeochemistry across the Great Lakes, among other things. This research has unveiled the lake's microbial diversity and provides a baseline for assessing future ecosystem changes. So we're very honored and excited to have her here with us today to tell her more about her work. Without further ado, Dr. Coleman.
Okay, can everybody hear me? Are we on? Louder? Okay. All right. Hopefully everybody can hear me on the webinar too. I've never done a remote webinar, but I think this is awesome. It's a great way to get people to attend seminars without actually having to fly there. I took the train, so yeah, this is great. Okay, so thank you for inviting me. I have only been to Laurel once for a workshop 5 years ago for CSMI planning, and I would really love to work with folks in this room a lot more because obviously you all are the experts on this ecosystem. I was trained as an oceanographer and then I moved to Chicago, and so I thought, why not study this inland sea that's right outside my back door? And it turns out there hasn't been very much microbial work done, and so it seemed like a great opportunity. And that's why I'm here today. So I'm going to give you a pretty broad overview of a lot of the microbial diversity work we're doing in the Great Lakes. There's lots of stuff I don't have time to talk about, but feel free to ask me later if you're interested. Okay, so why should we care about microbes at all? If you aren't a microbiologist, you might think, who cares, right? And I hope that this slide convinces you that microbes are really important. And so microbes, even though they're really tiny, do some pretty amazing things to alter our planet's surface. And so one thing they do is fix CO2. Is this mic better? Should I just stand here? Is that better? Yeah, okay. I feel like this was too variable. I'll just stand here. So as you are probably aware, of all of the carbon dioxide fixation that happens on Earth, about half happens through trees and plants in terrestrial areas, and that we can see and appreciate. But almost half of it happens in aquatic systems, primarily the oceans, and that's all microbial for the most part. And so we might not appreciate this, but some people would say that 1 out of every 2 breaths of oxygen you take came from microbes in the ocean, and the other one came from trees and plants and shrubs. So they've altered our planet's atmosphere on a really monumental scale. Another thing they've done is essentially oxygenate all of Earth's atmosphere through Earth history. So for 3/4 of Earth history, there was no other life form on the planet except microbes. And so this plot in the bottom left shows you the concentration of atmospheric oxygen throughout Earth's history. And for a long time, there was none until the cyanobacteria invented oxygenic photosynthesis. And that's the reason we're here today and breathing oxygen and that animals can exist. So again, 3/4 of Earth history, all microbial, but it was not boring by any means. The plot on the upper right shows phosphate profiles from the ocean, and this is just to show you that microbes are responsible for really large-scale patterns in geochemistry that we can go out and measure. And so we often see this drawdown of phosphate in the surface waters, and that's due to microbial activity. So again, another macro-scale pattern that's attributable to tiny cells that we can't see. And finally, as you're all aware, harmful algal blooms. This one happens to be from the Finger Lakes, where I grew up. But of course, you're very familiar with them in Lake Erie as well. And again, really massive consequences from microbial metabolism. So geochemists for a long time have recognized microbial metabolism because microbes are, as I said, fixing carbon, they're producing oxygen, they're also transforming nitrogen in a lot of ways that are really important, fixing nitrogen from the atmosphere, making it available for the rest of us to use. Only microbes do that process of nitrogen fixation. They do a lot of transformations of sulfur species. So sulfur cycling is really important, and that's primarily microbial. They also transform metals and toxins, pollutants, contaminants, everything. So they're performing these really essential functions that really touch on all of Earth's biogeochemical cycles, not just the carbon cycle or the nitrogen cycle. So at the same time, we've been undergoing this revolution in molecular microbiology, and now we can sequence DNA very cheaply and readily. And so we have, through this tool, essentially redrawn the tree of life. And so this is one of the latest iterations of the tree of life. And we've added a whole bunch new phyla, branches on here, complete clades that were not known to exist before, and this is all because of DNA sequencing technology. And so now I would argue the challenge is, how do we link these two? How do we link these biogeochemical processes that we know are happening and we know are microbial with this incredible molecular diversity that we can now measure very readily? And this is where it gets really challenging. And so I'm going to talk today about a lot of molecular data, and I will freely admit that it's really challenging to begin to link that with biogeochemical processes, but that's really where we're trying to go. So some of the basic questions we're interested in are, why is there so much microbial diversity on Earth? And what are its consequences? Does it matter that there's so much diversity? How did it form? What is it responsible for? How would the Earth look different if there was much less microbial diversity? So we are tackling this in the Great Lakes, and we think that's a really great model system for answering some of these questions. Of course, the Great Lakes are also really useful and important and valuable in their own right, but I don't think I have to explain that to this audience, so I'm not going to spend a lot of time telling you how important the Great Lakes are. You guys already know that. But I will tell you a little bit about why they're interesting from a microbial ecologist's perspective. So my lab is interested in understanding the rules that govern biodiversity and biogeochemistry. And so we're interested in things like resources, abiotic stress, temperature, pH, the availability of inorganic nutrients like nitrogen or carbon. We're interested in biotic processes like predation, viral infection, and even interspecies interactions between bacteria. And we're interested in how these three types of processes interact to shape the diversity and biogeochemistry, in this case of the Great Lakes. And so the Great Lakes are a really useful laboratory for studying this, and that's because they encompass such a wide range of conditions. If you're a microbe, the temperature changes dramatically, as you know. The light level changes dramatically. These are really deep lakes, which is actually fairly unusual. A lot of the lakes that are really well studied are really shallow, and so we don't have these vast volumes of dark oxic water like we have in the Great Lakes. They're also interconnected, as you know, and so that's really interesting from an ecological and evolutionary perspective, because it means that microbes can disperse from Lake Superior all the way through into Lake Ontario. But along the way, they're going to experience a whole range of conditions that may or may not be suitable for that particular organism. And so it's a really interesting dynamic system for looking at the interplay between physical processes like dispersal, geochemistry, and biological processes, and how these all interact and shape the microbial communities and functions. Okay, so again, this is not something I need to belabor to this audience, but the Great Lakes are also a really great system for watching change happen, perhaps too rapidly, but it's really important that we have these time series studies to really watch how things are playing out, basically in real time. And so a lot of this work, of course, was done right here by Gloral folks. So I'm just going to highlight two changes that I think are really important for microbes in particular. And one of them, of course, is the invasion of dracaenid mussels. So this is from a Mark Rowe paper. I saw him here somewhere. There he is. And so, as you know, and this has been updated, sorry I haven't updated it with the 2015 latest data, but as mussels have invaded Lake Michigan, what they've done is essentially reduce the spring bloom or completely remove the spring bloom of phytoplankton. And there's been a reduction in chlorophyll in the spring and in primary production. And so that whole base of the food web has been totally reorganized, transformed by these mussels. And as a result, we have this smaller spring bloom and potentially also a shift towards smaller cells. And this will become important later on because I'm going to talk about cyanobacteria. And the cyanobacteria I'm going to talk about are not microcystis. They're not toxic cyanobacteria. These are things like Senecacoccus that we think might become more important proportionally for primary production as the size structure shifts towards smaller cells in this more oligotrophic environment. Another obviously really important change is changes in ice cover and summer surface temperatures in the Great Lakes rising really rapidly. And microbes are really exquisitely tuned to their environment in a lot of cases. And so this again is a really important driver that we think could be changing community structures on an annual, interannual basis. Okay, so to understand the microbial diversity of this system and how this is affecting biogeochemistry and vice versa, we've been lucky enough to go out with the EPA and the GLINPO staff since 2012. So we started a time series about 8 years ago now. We're gearing up to go out in April as well. And so we've been on the Lake Guardian every April and August. And so, as you're probably aware, this time series goes all throughout all 5 Great Lakes. And so we've got good spatial coverage across all of the Great Lakes, although these are primarily offshore stations. We don't do a lot of nearshore sampling, so that's clearly a hole in our data. We also sample throughout the water column. And so just as a schematic, the 4 main depths that I will talk about today are surface, the deep chlorophyll layer, if there is one there, the mid-hypolimnion, and something near the bottom. B10 is 10 meters off of the bottom. So we have vertical structure of microbial communities as well as a spatial structure across all of the lakes. And we've got at least samples going back to 2012, although we've only analyzed a portion of that so far. Okay, so sampling for us is pretty simple. We haven't done any benthic work. This is all water column. And so basically we're taking the rosette and we're collecting all of the cells onto a filter, a 0.2 micron filter. And what I'm going to show you today is actually the very smallest bacterial cells. So they've been pre-filtered through a GFA. So that's a 1.6 micron cutoff filter. And so these are not going to be cells that are attached to larger particles. And clearly those are also very, very important. They're just not part of this data set that I'm going to show today. Okay, so here's just a brief outline of some of the topics I'll touch on. The first one, just at a very basic level, how does community structure change across the Great Lakes? And what do we think the drivers of that might be? What are the forces that are really shaping these communities across these 5 lakes and across depths? Second, I'm going to dive in a little bit more to one functional group that we're really interested in, and that's nitrifiers. So nitrifiers are a class of organisms that transform reduced nitrogen in the form of ammonia, urea, possibly cyanate, into oxidized nitrogen in the form of nitrite and then nitrate. So these are, we think, really active and important in the Great Lakes, and I'm going to tell you something about their diversity genome-wide. And if I have time, I will also talk about cyanobacteria as another case study functional group that we think is really important in the Great Lakes and that we've done a lot of genome work on. Okay, so to start with community structure, just at a very high level, who's there and what are the patterns across the Great Lakes? Because when I started this in 2012, this information was not out there. So there had been, bits and pieces from particular lakes. There had been a lot in Michigan. There had been some studies that did several lakes at once, but I think we've systematized it in a way that now we've got every lake sampled in the same framework. And this work was led by my postdoc, Sarah Paver, who was also a Sea Grant Knaus Fellow, for those of you who are affiliated with Sea Grant. Okay, so how do we measure microbial community structure, right? If you were measuring animal community structure or plant community structure, you'd go out with a guidebook or your vast knowledge, and you would do an inventory of all the species based on their morphology, right? And this very classic microscopy image on the right here from Jed Furman shows what a microbial community looks like under the microscope. This is from a natural marine community. A freshwater community would look very similar. There's not a whole lot there that you can tell. You can probably tell there's a diatom, there's some larger particles, there's a lot of smaller particles, and then there's even more smaller particles. And so from this, we can't say anything about the species that are there, right? As we like to say, microbes are morphologically uninteresting for the most part, right? And so we need a different approach to try to do a survey of which species are there. And the tool we use for that is this biomolecule called the 16S ribosomal RNA. And this molecule is universal. We all have it in all of our cells. And it forms a key component of the ribosome, the translation machinery that all life uses. And so by sequencing this very conserved marker molecule, we can essentially reconstruct the tree of life. We can put humans and bacteria all on the same tree with plants and animals and fungi. So that's what we use for a tool to do community profiling. And so we essentially go out, collect all cells on a filter, use PCR to amplify this molecule, 16S, and then sequence everything that's there. And that gives us essentially a readout of who's there in terms of species composition. And so the data essentially looks something like this. We get tables of numbers, and those numbers, the values are relative abundance. And so all the values for abundance here are going to be as a percentage of the total community. So everything all together adds up to 100%, and each of these individual gray squares tells you how much relative abundance there was for a particular taxon. So each row here is a different species or taxon, and each column is a different sample. And so we've clustered these together to put like with like, and some patterns immediately pop out. You can immediately see some groups of things that behave similarly. So there's a big group here that is enriched in these samples that all came from the deep bottom water samples. So these taxa seem to prefer deep water, and you don't find them very much in the surface water. This one on the top here, I'll draw your attention to, this CL511 is a group of chloroflexi that Vincent Deneff's group has actually studied quite a bit. And they are extremely abundant in those bottom water samples, but we hardly ever find them in surface water. So there's a clear ecological preference there for deep water. And then on the other hand, you can imagine there are things that really prefer the surface water, right? And so you can see some of these down here. There's a green star next to Synechococcus, which is a cyanobacterium. And so cyanobacteria, of course, need light for their metabolism. And so it makes sense that they would be enriched in surface samples and very rare in deep water samples where there's no light. Okay? So this is essentially what our data looks like. And so there's all kinds of interesting things embedded in here that I don't have time to go into, but you can see some specific examples here I've pulled out. And so the one on the left is this chloroflexi that we know from Vincent's work and some others is really abundant in deep water. And this is just a depth profile. Each colored circle represents a sample from a different lake. And you can see that at the surface, there's practically none of these cells, and they increase with depth. And this one next to it is interesting. This LD28 methylopumilus has a similar pattern, but we actually don't know exactly what it's doing. And so we don't know why it's enriched in the deep water. I should say the same probably applies to chloroflexi. We don't know precisely what it's doing in the environment, but perhaps we can get some clues by looking at these ecological distributions. Clearly there's something in deep water that it's eating, or there's something in surface water that it's avoiding, but there's some reason that you see these very strong gradients. And the two on the right show the inverse pattern. So Synechococcus, I mentioned, we would expect that to be really abundant in the surface water because it's photosynthetic. But this one next to it is not photosynthetic as far as we know, but it might be an organism that depends on phytoplankton or on cyanobacteria or on some product produced in the surface. And so, again, maybe we can start to get some clues as to why they have these particular patterns. So if you put all of that data together and look at it in another way, here I'm showing essentially a PCA type plot, a dimension reduction plot, where we've taken those big matrices of species abundance and reduced them down to two axes here that explain most of the variability. And what you can see is that these yellow points mostly cluster on the left. And these are, each point represents a sample and the microbial community we sequenced there. And these yellow ones are all more similar to each other than they are to the darker colored ones that group towards the right. And the yellow points represent surface samples. In the middle here, you can perhaps see some green dots. So those are from the DCL, typically the intermediate depth. And then there's mid-hypolimnion bottom samples off to the right. And so from this, you can right away see that there seems to be a pattern with depth. And the overarching factor that seems to drive these communities is depth. Samples from the surface look more similar to each other. Samples from deep water look more similar to each other, and those are quite distinct from each other. And you could see that from the data matrix heat map that I showed you initially. This is just another way to represent that. And of course, this makes sense because these are from the summer. These are from the August cruises on the Lake Guardian, when the water column is typically stratified at all of our stations. And so on the left here is just some environmental data to remind you of what that stratified water column would look like. And so we typically have a very shallow, thin mixed layer sitting on top of some colder, denser water. We've got a chlorophyll max. On the left-hand panel, you've got temperature and chlorophyll. And on the right-hand panel, you've got nitrate plus nitrite, so you can see there's patterns with depth there as well. So if you're a microbial cell, it makes a lot of sense that depth would be really important to you because we've got these really stark changes as you go down the water column. This was not unexpected, but it's always good to validate things that you think should be true. But we were also really curious whether there were any patterns across lakes, right? So is Lake Erie different from Lake Superior? Well, obviously yes. And so how do we see those patterns? So those patterns start to come out when you control for depth. Depth is such a primary driver that you don't see the lake-to-lake differences very clearly until you focus on one depth. But if we zoom in on just the surface across all of the Great Lakes, then it becomes very apparent that there are lake-to-lake differences. So this is another one of those plots, and now it's just surface samples. And now you can see that there's clustering that happens between upper lakes and lower lakes. And so the samples are most similar when they come from either Erie and Ontario, are similar to each other, compared to Superior, Michigan, and Huron. And again, that seems sort of intuitive, right? And so what's really interesting is some of the individual taxa. You can pull out really interesting patterns. There are upper lake specialists, lower lake specialists, but then some weird things that we don't quite know how to explain, like this one on the bottom is a flavobacterium, and we only find it to be very abundant in Lake Superior, and then for some reason we don't find it anywhere else downstream at very high abundance. So there's something that the microbes are sensing about these individual lakes and their chemistries, also influenced perhaps by dispersal, where they're coming from, the source, and this is what we're trying to understand now, what generates these patterns. So again, if we were to reduce this to a PCOA type plot and just focus on individual depths, so the one on the top is just surface samples. So each dot is one particular sample from the surface. And now we've got 4 years worth of data. And they're color coded by lake, which matches the map up in the corner. And so immediately, hopefully you can see that the reds and oranges are sort of down in the bottom left corner. And the purple, blues, and greens are in the upper right corner. And that's essentially, you could draw a line down the middle and separate the upper lakes from the lower lakes. So I think that's pretty cool. and probably has something to do with their productivity, their temperature, nutrients. There's a lot that covaries, obviously, so it's hard to pinpoint one particular factor. But what I also find is really interesting is you can also see it in bottom samples. And I expected the bottom samples to be a little less lake specific, perhaps more uniform across lakes. But in fact, you can still see clustering across the bottom samples. And now, if you'll notice, there's a couple red points that have grouped over with the upper lakes. And those are probably deep samples from Lake Ontario. And this is something we see repeatedly, that Lake Ontario is a really interesting hybrid. It gets a lot of its surface community from Lake Erie, but the deep waters begin to look a lot like the upper lakes, especially as you get further east into Lake Erie. So I think Lake Ontario. Lake Ontario is a really dynamic environment, and it's where the communities are really sort of shaking out. They're sorting themselves out, having been disturbed by this transit through Erie, and they're sort of gradually perhaps beginning to resemble the upper lakes more. Okay. So this plot sort of summarizes things in a slightly different way. And in this plot, every particular dot is a taxon, a particular species, if you will. And what I'm plotting on the bottom is observation frequency, and that's what fraction of our samples had this particular species. And on the y-axis is its relative abundance, and I think it's maximum relative abundance in any given sample. And so anything that's over on the far right-hand side on this 100 line, those taxa were all ubiquitous. So every single sample we ever sequenced had representatives from these taxa. And these are common freshwater taxa that you would find in Lake Mendota, you'd find them in Lake Baikal, you'd find them in freshwater around the world. These might be slightly different sequence variants, but they're certainly related to these widespread freshwater taxa. So there's a few ubiquitous things that are on this line, and they tend to be pretty abundant. There's also a lot along the bottom, right? So lots of things that are sporadic. They're in a few samples, but not others. And they never reach very high abundance, maybe 1% at their max. So there's a lot down there on the axis, right? And then there's a few in the middle that are sporadic and sometimes reach high abundance. And I'll call your attention to one because we're going to talk about it next, and that's nitrifiers. And so in the middle here, there's some dots labeled nitrosospira, nitrotoga, nitrosomonads, nitrosoarchaeum. Those nitro prefixes are a good clue that it's a nitrifier. And this pattern was really interesting to us because it suggests that they're bloomy in a sense. They sometimes reach high abundance, but not in every single sample. And that's something we will come to next. So the take home from this, I think, is that depth is really the strongest driver of the communities in the Great Lakes. And when you focus on a particular depth, you really begin to see lake-to-lake differences. And most taxa are rare, right? There's a few that are ubiquitous and are everywhere, and they're really good at perhaps tolerating and thriving in a variety of conditions. And then there are some that are quite specialized, and we only see them in particular instances. Okay, so now I'm going to dive in a little deeper. This was sort of the high-level community view, and I'm going to look at one particular group in particular, and that's the nitrifiers. And this is the work of my student Justin Podowski, who's pictured here. And we're going to look at their abundance and diversity across the Great Lakes, and also we're going to dive into their genomes and see what those genomes might possibly tell us about ecology and biogeochemistry. So the 16S ribosomal RNA, which all the data I just showed you was from 16S, is a really useful tool, but it also has its limitations. And it doesn't tell you anything about what's going on in the rest of an organism's genome. And so you can have two kinds of bacteria with exactly the same 16S ribosomal RNA, but functionally they could be quite distinct from each other because they could have variation elsewhere in their genomes that we can't see by 16S. So it's really hard to go from a 16S sequence to a functional prediction for what an organism is doing in the environment. And so to get closer to that particular question, we use metagenomics. And metagenomics is simply shotgun sequencing, for those of you who use that, chopping up all of the DNA in a sample and attempting to reconstruct genomes and organisms and functions from this complete mash of DNA. So unlike 16S, where we were looking at one particular gene, now we're looking at every single gene in the genome simultaneously, every single organism, and we're trying to make sense of it. So it's essentially an exercise in putting puzzles back together. Imagine taking a whole bunch of 1,000-piece puzzles, dumping them all in the middle of the room, and then trying to put them back together from this big messy pile. That's what Justin does for a living. So what Justin has done is actually quite successful. So from that messy pile of pieces, he has reconstructed over 750 genomes. We call these near complete. They've got over 70% completion. And so this diagram, I don't expect you to get a lot out of it, except it looks pretty impressive, right, for Justin's work. And so we've got a phylogenetic tree in the middle, and each particular tip on that tree is a genome that we've reconstructed from the Great Lakes. And the rings on the outside are colored by the Great Lakes, according to that map. And so where you see a bar that's dominated by yellow, for instance, that's a genome that we found essentially only in Lake Erie. And some of them are pretty evenly split among all 5 lakes. And so those would be cosmopolitan species that we see in every sample across the Great Lakes. So again, we could spend days looking at this, but we're going to zoom in on just a couple particular groups today. So in my lab, we work primarily on these 3 groups: the nitrifiers, cyanobacteria, and another group of phototrophs that I'm not going to talk about today. So let's start with the nitrifiers. So nitrification, as a lot of you probably know, it's a key flux in the global nitrogen cycle. And it's also a microbial energy metabolism that's really important in dark water. So at the surface, there's primary production from phototrophs. And in deep water, nitrifiers are able to fix CO2 through this nitrogen-based metabolism. And so what nitrifiers do is take reduced nitrogen. So here we've got ammonium on the left, or urea and cyanate. And this top group of organisms is called the ammonia-oxidizers. And so those take this first step, ammonia, to nitrite. And then there's another group of organisms, the nitrite-oxidizers, that takes that nitrite to nitrate. And through those successive oxidations, the cells gain energy and can fix carbon. Just to note, recently there's been an organism described that can do this whole process. That's shown on the bottom here. We actually don't find those in our samples in the Great Lakes. So we do not find Comomox, at least in the water column. So nitrifiers are phylogenetically really diverse, and we didn't really know much about who the nitrifiers in the Great Lakes were before we started this work. So the nitrifiers fall into proteobacteria. This is just a tree of life, all of the phyla of Bacteria, Archaea, and even eukaryotes, based on the Laura Hug tree that I showed previously. And so I've colored the clades that have nitrifiers. And as you can see, they're pretty scattered throughout the tree, including a bunch of proteobacteria, the phylum Nitrospirae, the phylum Nitrospinae, Chloroflexi, and even Archaea, the Thaumarchaeota. And so one of our first questions was, what are the taxa in the Great Lakes and how are they distributed? So what we first found was that nitrifiers seem to prefer deeper water. And this was not a surprise, because this is what's been found in the oceans and in other lakes as well. So this plot, each square or circle here is a particular sample, and I'm showing the total abundance of nitrifiers as a percent of the community. So they get up to about 10% of the community in a lot of deep samples. And this is depth, or PAR, photosynthetically active radiation on the y-axis. And so near the surface, where there's high light, we don't see hardly any nitrifiers. But as soon as you get down below this 1% PAR line, then we see nitrifiers become particularly abundant. And it's not entirely clear why this is the case, whether the nitrifiers are actually directly inhibited by light, or whether the competition is just too strong for ammonium in the surface water with phytoplankton and other organisms. And we also find that these nitrifier taxa are spatially variable across the Great Lakes. And so here I'm showing the map with all of our master stations highlighted. And for each master station, I'm plotting the abundance of 2 different kinds of ammonia oxidizers in green and 2 different kinds of nitrite oxidizers in pink. And so on the left-hand panel, you can look at the ammonia oxidizers. So we've got archaea and bacteria. And ammonia-oxidizing archaea are really abundant in the oceans. They're the dominant ammonia oxidizer in the oceans. In the Great Lakes, they're a little bit more sporadic from our data. We find them in Lake Superior quite a bit on the left. We find them in Lake Ontario, in Lake Michigan at some stations, but we find virtually no archaea in Lakes Erie and Huron. And in the right panel, the nitrite oxidizers, you can perhaps see that there's a little bit of switching happening. The dominant nitrite oxidizer switches between a filled pink circle and an open pink circle. And those are just 2 different versions of nitrite oxidizing bacteria. But this was really interesting to us because, you know, on the one hand, it looks just like a big, well-mixed lake. You know, even within a lake here, though, we're seeing switching between stations. So what is it about, for instance, the central basin of Lake Michigan that encourages a different assemblage of nitrifiers than you see in the northern basin or the southern basin? And so it tells us that the microbial community is changing at a very fine scale, perhaps, in space, and that the geochemistry is probably changing, and that's what they're sensing, right? There's something different about that environment that is selecting for one nitrifier versus the other. And so, in a way, the microbes are reporting to us what they're feeling out there in the lakes and where conditions are different from their perspective. And so that's what we're trying to understand. If we could understand what they're saying, they might say, hey, there's more urea at this station, or hey, there's less predation at this station. So those are the things we're trying to get a sense of from their genomes. Okay, so how do we do this? What do the genomes tell us? So as I said, we've reconstructed genomes for all of these taxa. And so we can look at the functions that are encoded in those genomes, look at their gene content, and ask what's different and what might be driving these differential distributions across the lakes. So if we first focus on just the ammonia-oxidizing bacteria, we find some really interesting patterns. We were able to detect 4 different types that are highlighted in green. We call them very creatively type 1, type 2A, 2B, and 3. And you can see immediately that their genomes are distinct. So the type 1s have smaller genomes and lower GC content, meaning their genes, the DNA is more enriched in A and T bases and less enriched in G and C bases. And typically that correlates with genome streamlining, with low nutrient conditions, and this is something we've seen in many environments. So from this, I would predict that type 1s are going to prefer the more oligotrophic stations out of our data set. We can look at, for instance, their carbon fixation pathway. So as I mentioned, these are primary producers. These are fixing carbon by the Calvin cycle. And so it turns out they have different forms of rubisco. This form 1AC, 1AQ, and 1C, these are different kinds of rubisco, the enzyme that's doing that carbon fixation. And so we don't necessarily know what that means right now, but it's interesting and suggests that perhaps this diversity could influence things like primary production, right, in deep water. We found that in one case, the type 1s, they encode an enzyme called urease. And that's really interesting because it suggests that they're not relying simply on ammonium as their source of reduced nitrogen. They can perhaps access nitrogen through urea. How much urea is out there? We don't have really great data on the spatial concentrations of urea across the Great Lakes, but this suggests that at least in some places, urea is a really important source of nitrogen. And here's where it gets really interesting and surprising. These type 1s, perhaps the most oligotrophic adapted of the group, also encode this gene called proteorhodopsin. And that's a surprise because it involves harvesting light energy. And I recently told you that nitrifiers prefer dark water. So why on earth would they have this light harvesting gene? We'll get to that. Here we go. So light harvesting proteorhodopsin is diagrammed schematically on the left. This is a protein that crosses the cell membrane, and it is attached to a chromophore called retinal that absorbs photons of light, and it pumps protons outside the cell. And that's useful for the cell because then it builds up this gradient, and those protons can come into the cell and do useful work, like make ATP, run a flagellar motor, help transport nutrients across the membrane. And so this is a way that we think bacteria might make extra energy using sunlight. But it's surprising because, as I've shown here in the upper right, just to remind you, we find these organisms in deep water where there's typically not any light. And so I don't actually have the answer. I don't know why they have this, but one hypothesis is that given the mixing timescales of the lakes, maybe they are mixed up to the surface frequently enough that they have an advantage there when they have this light harvesting proton pump, and perhaps there's not so much ammonium around. Maybe it's a way to wait out starvation conditions when ammonium is not available or urea is not available. But now we're trying to get these guys into culture in the lab so that we can understand how this proteorhodopsin is regulated, how it functions, and why it's beneficial for the cells. And this alignment, if you're a proteorhodopsin expert, shows that our nitrosospira proteorhodopsin has all of the conserved features that you would expect in a proteorhodopsin. So at least based on its sequence and based on the fact that this genome also encodes retinal biosynthesis, we think this is real. We think it's really functioning as a rhodopsin, a proteorhodopsin, but I can't tell you what it's doing. To be continued. So we can do the same thing with nitrite oxidizers. And again, we've got two groups here in pink that are from the Great Lakes. And again, they're different in their genomic content. And as a result, they have different distributions across our stations. And we're trying to understand what this means, and we've got some experiments planned. But for instance, the top one, Great Lakes nitrospira, encodes two genes, cyanase and urease. And those two genes suggest that it can use cyanate, which is not something we talk a whole lot about in aquatic systems, or urea as nitrogen sources. But what's interesting is that these are nitrite-oxidizing bacteria. These aren't the ammonia-oxidizing ones. These are doing the second step of the pathway, not the first step. And so why would they have genes for degrading urea and cyanate when they actually need nitrite? And so one hypothesis in the literature, and this has actually been demonstrated in some marine cultures, is that there's cross-feeding happening between the ammonia-oxidizing and nitrite-oxidizing organisms. So in this case, it would mean that the nitrite oxidizer is able to cleave ammonium from cyanate or urea and pass that ammonium to the ammonia-oxidizing partner, who then would feed the nitrite oxidizer nitrite. Whether this is actually happening, it's hard to say. It's hard to say how this would, imagine how this would happen in a really dilute environment that's not attached to particles or biofilms or flux. It could also be that these nitrite oxidizers are just using that reduced nitrogen for their biosynthetic needs, because every cell needs nitrogen to make proteins. So it's just a really interesting potential ecological story that could come out of this. Okay, but going forward, this genomics is all great, but how does this community structure affect nitrification rates? That's a really big open question. There have been a few nitrification rate measurements in the Great Lakes, but not a whole lot. Most of those have been in Lake Superior. And how does the physiology differ among these nitrifiers? Why do we have multiple kinds of organisms doing essentially the same process? It doesn't matter for rates of primary production or nitrification, and what in the environment is selecting for these distinct assemblages. Those are things that we're trying to answer now. Okay, so I've got a little time so I can talk for maybe 5 minutes about cyanobacteria. I want to make sure I leave enough time for questions. But we can essentially do very similar things with cyanobacteria and ask, what is the diversity of cyanobacterial types out there in the Great Lakes, and what does it tell us about their ecology and perhaps biogeochemistry? And this work is led by Sarah Paver again and Justin Podolsky. So this data is actually 16S data, which gave us a first clue that there might be something interesting happening with cyanobacteria. And what we found is that there are a whole bunch of different Synechococcus that are coexisting across the Great Lakes. And this plot is just to show that each of these OTUs, each different cyanobacterial sequence variant, has a different distribution across samples. So some are enriched in the upper lakes, some are enriched in the lower lakes, some of them are cosmopolitan. And so this gave us a clue that maybe there's some really interesting dynamics and diversity there. It's not a uniform population of Synechococcus. So we reconstructed genomes just like we did for nitrifiers. And so this slide puts them all in a very big, messy tree. But if we just walk through a couple of the highlights here. So this tree actually is constructed from all kinds of Synechococcus, many of which are actually oceanic, marine strains. And what's interesting to me as a previous oceanographer is that the freshwater and marine strains are quite closely related. There is not simply a deep divergence between marine and freshwater. It seems to have happened a couple times, perhaps. There have been several transitions. And we find a little bit of intermingling. And so that's a really interesting evolutionary observation. And so the ones we've sequenced from the Great Lakes are identified here by GL1, here's GL4, GL2, GL5, GL3. So those 5 groups are all genome types that we reconstructed from the Great Lakes. And as you can see, they're really broadly distributed throughout this phylogenetic tree, meaning we've got quite a diversity of Synechococcus in this system. And if you look at just the next column, it says pigment type. Oh, sorry. And they've got symbols here. And each of those symbols represents a pigment type. And what's known from marine Synechococcus is that they come in all different flavors and colors. And they have different suites of pigments that are typically adapted to that organism's niche in the water column. So cells that live further down in the water column have a different set of pigments than ones that live close to the surface to match the light field that they would experience at depth. And similarly, coastal strains and open ocean strains have different pigments because the light quality and composition is different. And so based on these marine pigment types that have been defined, we can assign our Great Lakes ones to pigment types. And we basically find all of the pigment types. So this GL5 down near the bottom has a green square. That means it's a type 1 based on its genome. And it's predicted to be green in color and not have any phycoerythrin. We've got purple circles here for 3 of them, and so those are predicted to be type 2. And so in culture, we'd predict that these would be sort of pinkish with plenty of phycoerythrin. And then this GL1 up on top is predicted to be a type 3, and that could be some shade of orange, orangey yellow, pink that we don't know yet. So this is interesting, and it suggests that perhaps these different varieties of Synechococcus are partitioning in the Great Lakes based on the availability of light and what that light field looks like to them. So we can see this partitioning if we look at the abundance of these 5 types. This is, sorry, a pretty complicated diagram, but if we just look at lake up in the top left, panel A, one thing that should immediately jump out is that the green bars, GL5, seems to be an Erie-specific type. That one we only find in Lake Erie. The upper lakes are dominated by this GL3 type and these GL1s. You can see this perhaps partitioning by season, by depth, by any axis that we look at. It's a non-random distribution of these 5 types. And so from this, we can start to make predictions about their ecology. And so GL1 seems to be specific to the upper lakes. GL2 seems to really prefer colder water, the DCL and spring. GL3 is everywhere, cosmopolitan. This blue one, GL4, seems to prefer Erie and Ontario, the lower lakes, and GL5, as I said, was Erie specific. So already we're getting some hints as to how this diversity is mapping to the lakes. And so finally, one thing we're doing now is attempting to connect this genomic information with phenotypic information in the form of pigments. And so one of the really interesting ways to do this is using high-resolution flow cytometry. And so my collaborator, Ann Thompson, at Portland State University, has a really amazing flow cytometer with 5 or 6 lasers, many detectors, and many years of experience doing this. And this is example data from marine strains. These were from cultures. But what Anne was able to show was that using combinations of lasers and detectors, you can essentially differentiate the different pigment types of Synechococcus based on their flow cytometry signature. And for those of you who aren't familiar with flow cytometry, there's a diagram here in the middle showing essentially a single stream of cells going through a series of lasers. And as those lasers hit those pigments in each cell, they will emit these fluorescent signals that you can capture on detectors, and those cells can then be characterized by this set of signals. And so what Anne showed is that type 1s and 2s and 3s could all be differentiated by using this combination of lasers. And so that's essentially what we're trying to do now in the Great let's. And so this is actually samples that Anne has processed for us. These are from the Great Lakes. And she's able to detect, 7 different varieties of picocyanobacteria using her multi-laser method. And this is not from one sample. I should say this is an overlay of several samples. So this is sort of the cumulative diversity we see across several lakes. Typically in one lake or one sample, we would only see 2 or 3 of these populations at any given time. But together we see 7 throughout the Great Lakes. And so what we would like to be able to do now is begin to map them to their genomes. Now that we have this genomic information, we have this flow cytometry information, can we start to link the two? And from her past work, it looks like you could separate these types based on the diagonals. It looks like we've got perhaps these type 2, 3b, 3c resolved here, but that remains to be confirmed. And the way that we're doing that is actually, this flow cytometer is also a cell sorter. So we can sort individual populations of cells into plates or tubes and try to sequence those, grow those up, do further work on them. So now we're trying to separate essentially each of these populations and begin to map it back to its genomic information and its ecology. So based on flow cytometry, Anne has basically done a similar exercise to what I've done and map these different pigment types across lakes and samples. That's what's shown along the x-axis here in different samples. And so now she can tell from her data where these different pigment types are most abundant. And we can already start to make some predictions about which one's which. So the GL5s I mentioned were the type 1s that lack phycoerythrin. And so lack phycoerythrin, these PE minus ones. And there's one that's Erie specific. So that's probably the GL1. So we can start to map these and we'll be able to do this more concretely in a few weeks when we've got some more data. So again, we're left with the same questions. We've sort of mapped the diversity at this point, not only at 16S community level, but also within a particular functional group. But now we really want to know why and how. So how did it get there and why does it matter? Does it matter? So how does community structure affect primary production rates? Do we get integrated primary production that's greater when we have this diversity of coexisting types than we would have if we had just a single clonal population of one type throughout the water column? And how does their physiology differ? So we're trying to isolate each of these types in culture so we can do physiology experiments and really figure out what's distinct about them. Okay, so just to summarize, this is sort of a picture of what we do in my lab. We do sort of a back and forth between lab work, which I didn't talk so much about today, and field work. And so obviously we've done a lot of work with the Lake Guardian, and this is a really core component. And from that, we can get genomes and we can generate hypotheses that hopefully we can test in the lab with model systems, cultured isolates, do some RNA-seq, some proteomics to understand their physiology, go back to the field, do incubation experiments, for instance, on board, and try to connect this loop. So just to summarize, we have found that deep lakes harbor a lot of novel phylogenetic and genomic diversity. Quite a lot of taxa we find have no close representative in any database, except now perhaps from Lake Baikal, there's some sequencing coming out. But before that, there were no known isolates for almost any of these deep water taxa. The ecological distributions that we see with 16S show us that the surface versus deep distinction is really important for microbes. And we also see upper versus lower lake preferences. And so going forward, we're hoping to continue this time series and see how these patterns change over time as conditions change year to year. This year, for instance, will be quite interesting because it was a fairly low ice year. It was a pretty warm year by comparison to what we've had. So it'll be interesting to have another data point that's quite different. We've got more than 750 genomes that reveal metabolic and ecological diversity that we didn't know much about before. Nitrification is active in the lakes, we think. We think it's really important, but it's not one uniform assemblage of nitrifiers. It's actually quite heterogeneous in space, and we don't know the answer yet as to what this diversity means in terms of rates, and that's where we're going next. And finally, cyanobacteria, we think, are probably becoming increasingly important in the Great Lakes, and they are incredibly diverse phenotypically and genomically. And again, how this diversity matters for primary production and for food web structure is something that we'd like to investigate more. So I'd be happy to take questions. I'm happy to talk more in the future. I would love to collaborate. And if there's anything in here that you think is interesting, then please get in touch and we'll do some more work. So thank you very much.
So let's take a question from the webinar real quick. And folks, if you want to ask questions, you can stay over a little bit, but we're going to end the webinar at 2. So we'll get to this webinar question. Christina Heinemann is interested in Maureen's thoughts on the implications of these research findings for lake management. She said, how about a t-shirt with the genome of the Great Lakes?
That would be awesome. Yeah, I have this this idea of constructing like a genome atlas of the Great Lakes where it's interactive, right? And you could click on a particular point and pull up all the genomes that come from that water. What it means for management, I think, is really still an open question. And from our perspective, we see all this diversity and it's really interesting. And it tells me that there is something different chemically, physically, biologically as we go from station to station in these Great Lakes. And I don't think we fully appreciate what those differences are. Like we can measure things as geochemists that seem relevant to us. We can measure light and we can measure pH and we can measure nitrate, right? But are we measuring the right variables is something that I don't think is clear. And I think the genomes can give us some insights into that. So we're trying to use the genomes to tell us what the microbes are sensing is different. For instance, we found some interesting patterns regarding metal transporters, metal utilization between the lakes. And there's not a lot of people measuring trace metals in the Great Lakes because that's really hard. But from microbial genomes, we're getting a sense that maybe trace metals are one of the selective pressures that could be important in distinguishing microbial communities across the Great Lakes. So I think we're still at the early stages. I don't think there's a clear path right now for how we would use this in management, but that's where we eventually hope to go.
Thank you for that. We have 2 more questions online, and then we'll get to the folks in the room, and we'll end the webinar at 2. Pardon my butchering of this word. What wavelengths of light does proteorhodopsin absorb? Have you looked to see if you could have enough of those wavelengths in deeper waters to grow while avoiding competition or more harmful shorter wavelengths at the surface?
Yeah, that's a really interesting question. And the proteorhodopsins are actually, in a way, like cyanobacterial pigments. They can be tuned to different light fields, different wavelengths. And so the one that we have appears to be a green-tuned proteorhodopsin based on what is known from the literature. And I don't know if that would be, if there would be sufficient light available at the appropriate wavelength where we see these. So now we're trying to do some experiments on the ship to do some light transplant experiments, move things from one light intensity to another, look at expression of proteorhodopsin throughout the water column.
Thank you. And one final question on the webinar. Thank you for a great talk. I'm curious if you measure net primary production or bacterial production to look at how communities might be impacting production.
We have not yet, but we just wrote a proposal to do that. So if NSF gives us some money, we would love to do that. But I do think it's really important now to try to link this diversity with actual process measurements, right? It's easy to take a snapshot of the DNA or the RNA, but measuring the process rates like primary production is sort of the next step, and linking those is really sort of the crux, the holy grail of all of this.
Thank you. That's going to be the end of our webinar. We're going to log out, but folks in the room, if you still have questions, feel free to ask them. I'm going to turn the microphone off.
Vincent.
I was wondering, like with the Synechococcus, you kind of showed like two major